So we have Dr. Sean Hoban with us today to present on conserving genetic diversity of the endangered Texas endemic big red sage, or Salvia pentstemonoides. As tree conservation biologist at the Morton Arboretum, Dr. Sean Hoban works to understand, document, and conserve plant species, both rare and common. Dr. Hoban has published 49 peer-reviewed scientific articles and presented his work at dozens of regional, national, and international conferences. He is also an editor for the journal Conservation Genetics. He has contributed technical expertise to several government agencies, including helping develop a new management policy in South Africa. Before coming to the Arboretum, Dr. Hoban received a PhD from the University of Notre Dame under Dr. Jean Romero Severson. He then worked as postdoctoral researcher in France and Italy, and as a fellow at the National Institute for Mathematical and Biological Synthesis in Knoxville, Tennessee. So thanks, Sean, and you're on. Although you have to unmute yourself.
Great. Can you hear me okay?
Yes, yes we can.
Okay, and you can see the screen okay?
Yes.
Great. Well, thank you everyone for coming today. This is a really nice opportunity to speak to a new audience, to tell you a little bit about the project that we performed in collaboration with the Texas Parks and Wildlife Department, and to introduce you to a little bit of conservation genetics. So our focal species is shown here, along with its habitat. And I'll talk about how we can use genetic knowledge to conserve this and other rare plant species. And this talk is co-authored with Mara Garner, who was a research assistant in our lab, and who I'll tell you a little more about later. First, I'd like to tell you a little bit about the Morton Arboretum, which is where I work. We are a living museum, a center of tree science research. We are also a park, a botanic garden, a place where people go to hike and bike. We have forestry plots, we have a tree breeding nursery. It's a really diverse place with a lot of functions. We also educate and inspire the next generation of botanists and plant enthusiasts. We have over 1 million visitors a year. We have partnerships with many communities throughout the Chicago region. So hundreds of communities where we help develop tree advocacy, tree care guidelines, local municipal policy about trees, preserving old trees, for example, green spaces, etc. We also mentor future plant scientists. So we have high school students, undergraduates, graduate students, postdocs, as well as retired people and other associates who work in our plant science research. So some of those are shown here. We've been around for almost 100 years. I've been working here myself for almost 5 years. We are located outside Chicago, about 30 to 40-minute drive west of downtown Chicago. Our total staff is about 200, and some of our staff members are shown there on the left. Our science staff is about 30 to 35 people, depending on grants and temporary hires, split among 11 research groups that cover all areas of tree research: phylogenetics, arboriculture, root science, soil science, forest ecology, community ecology, genetics, conservation, etc., as well as important science communication. So who am I? As Anna introduced myself, I received my PhD from Notre Dame, which is in northern Indiana, in 2010. I grew up in Louisville, Kentucky, and I lived there for most of my life up until that point. After I got my PhD, I lived and worked in France and Italy for a few years. And then I moved to Knoxville, Tennessee, where I worked at a math bio center located at the University of Tennessee, Knoxville. I'm a scientist, but I also have other roles. So I'm a journal editor. I lead working groups in conservation biology, and I work with the Center for Plant Conservation. Maybe some of you have heard of the CPC or Center for Plant Conservation. So I work with them to develop conservation guidelines. I'm also active as a seed collector and plant explorer. So I've been on several trips to collect seeds of rare species. I occasionally do teaching, outreach, things like this webinar, and I mentor students and other young researchers. Why am I here today? Why do I work at the Morton Arboretum? Why am I a conservation biologist? These are some of the things motivating me to do my work. Many plant species are threatened. About 20% of all plant species are threatened currently with extinction. Millions of populations and an important habitat are disappearing all the time. And only a small fraction of the planet and of species adaptive potential is protected either in situ, protected areas, reserves, or ex situ, seed banks or botanic gardens. The challenges for plant conservation are growing. We have every year more and more introduced pests and pathogens, including on some of our most common and well-known species, ash, chestnut, beech, and several diseases in oaks. Conversion of natural habitat continues, and climate change will make challenges for plant conservation even more acute in the coming years, and already are making it more difficult. In this context, these are the 4 areas that my lab group at the Morton Arboretum works on. Try to understand why species are threatened in the wild. Is it small populations? Is it habitat fragmentation or loss? Is it disease, etc.? We try to understand and predict how trees will respond to ongoing and future climate change. We also we work on measures of conservation, so measures of conservation success, the GSPC, that's the Global Strategy for Plant Conservation. So we try to measure conservation success to inform policymakers. And then lastly, we work on how do we do a better job as botanic gardens, seed banks, etc., for conserving species ex situ. What is that word ex situ? First, I should mention these are a few of my lab members shown here. I'll introduce them in more detail later. What does ex situ mean? This is outside of a species' natural range. Shown here are 4 really cool ways of conserving a species ex situ. Starting at the top left, that's what's called tissue culture. It's basically growing plants in miniature. Then cryopreservation, so that's ultra cold freezing. Then seed banks. Those are kind of obvious, small envelopes of dried seeds, usually kept in the cold. And then this is a picture of the Morton Arboretum, so living plants as representing the natural variation. Why is a botanic garden working so much on conservation? Well, it's consistent with our mission. These are 3 mission statements, excerpts of 3 mission statements from 3 globally well-known botanic gardens, the Morton Arboretum, the Royal Botanic Garden Edinburgh, and the Lady Bird Johnson Wildflower Center. And you can see that each of these gardens has conservation of plants for the benefit of people and the planet, especially diversity and native species, really central to our mission. Why do I focus on genetic diversity? In my laboratory and at botanic gardens and seed banks, we want to conserve the variation across a species native distribution. A plant growing in one place is different from a plant of that same species growing in another place because of local adaptations or other genetic differences. Genetic diversity helps species evolve. It helps them adapt to ongoing changes in the environment, changes in temperature, precipitation, what we call biotic interactions. So those are pollinators, herbivores, things like that. Genetic diversity provides breeding potential for horticulture or agriculture or forestry. Genetic diversity within species also makes ecosystems more stable. Lastly, genetic diversity is protected under numerous laws and conventions, including the United States Endangered Species Act and the Canadian Species at Risk Act. So this field of conservation genetics, why do we study genetic diversity? It can help us make decisions about plant conservation. So understanding the genetic diversity of a species helps us determine what populations to focus on. Are some populations more diverse or more distinct than others? Are some populations inbred, where they might be suffering what's called inbreeding depression, lower reproductive output because they're so small? How much genetic diversity do we have in botanic gardens and seed banks? And what are the most appropriate actions needed to help a species survive? There's really 3 phases of a genetic study, and I'll walk you through those today as we talk about Salvia pentstemonoides. First is the field work. This is where we go out and we collect leaf material from individuals in wild populations. We then study the DNA. So we bring it into the laboratory, we extract the DNA and we study it. And lastly, we kind of calculate results and interpret them. Those are the 3 main phases that I'll talk about today. What is this genetic diversity? What does it look like? What are the kinds of things we're studying in wild populations? Well, some of this might be a little complicated, but I'll walk you through what are some areas of genetic diversity. So hopefully you can see my mouse, but if not, you can see there's something called richness. This is basically the number of variants, the number of different forms or functions that exist in a population. Heterozygosity is how that's distributed among individuals. So if you remember your genetics class in high school or college, that's like big A, little a versus big A, big A. That determines things like eye color, hair color, all kinds of traits. We can also learn about how populations are different. Most species are divided into populations that occur in distinct sites, geographies, lakes, forests, etc. We can look at how many genetic populations there are and how different they are. So here, these are more populations on top and more different populations on top. And then lastly, we can understand inbreeding. So that's how much are individuals mating with each other. This can happen in plants, in some animals, and in some cases it's not a problem, but in some cases it can lead to abnormalities, dysfunctions in a plant. So I mentioned there's 3 main phases of a genetic project. Let me talk about that in a little more detail. So starting at the top left, this is field work. This is actually an oak, a shrub oak, growing out in Utah. I also collected this same species in Texas. We find the individuals in the field. We collect them. They all get unique ID numbers so that we can trace the genetic data back to the plant in the field at the end of the project. This is Bethany Zimwalde, who was in my research lab, and she's now a PhD student at the University of Florida. We get the DNA out of these leaves. So we take this tough leaf, grind it up, and use some soaps and detergents and other chemicals to separate the DNA from the rest of the tissue. We then use what's called PCR to examine that DNA, okay? And then we take that, we put it on this big machine called a DNA sequencer, and then we get something like this. This is the actual DNA sequence. All these letters here are the sequence of the DNA, and this is just a picture of it. Oh, this is here. We just check to make sure that each of these steps is working properly. And then in the bottom right is what the actual data looks like. This is a way of measuring actually the length of a part of the DNA. You see there's variation among each individual in that column, and that's what we call genetic diversity. So now let's talk about our subject at hand today, Salvia pentstemonoides, or big red sage. Starting in about the 1940s, this species had been proclaimed likely extinct. But it was rediscovered in the 1980s by happenstance by botanist Marshall Enquist. The species is shown here, both the full plant and a close-up of the flower. It has a basal rosette and a stem with the flowers on it that can be up to 5 feet tall. There are very few populations of this species known in the wild. I won't name particular sites or show them on a map just because of the sensitivity of this species, because it is so rare and threatened. There might be between 5 and 10 sites. Some have been revisited and plants have not been found. Others have been revisited and plants have been found. These populations are also very small. Some of them have only a few plants known. What's really interesting about this species is it's common in cultivation. So there are several nurseries in Texas and elsewhere across the United States where you can purchase this plant. So it's super threatened in the wild, but it's a beautiful plant and it can be found at some nurseries. If you'd like to read more about the rediscovery of this species, you can Google On the Trail of the Big Red Sage by Marshall Enquist. You can find this article on the internet. It's a nice little plant exploration and commentary about this species. Let me show you a few pictures of the habitat. These are some waterways of the kinds of places where you might find big red sage. So it's found in limestone-based rock, usually eroded rock along waterways. It's often found in pretty precarious places, which might explain part of its rarity. So you can see here it's along the bank of a stream, really subject to erosion. Here on the edge of a cliff, also very subject to erosion. Here, this is a stream that had recently had a really large flooding event, and this is just gravel and cobble that was deposited in this seasonal stream from a really large flood event. So the types of habitat where this is found are especially vulnerable just to natural changes, in addition, of course, to loss of habitat and other changes caused by humans. So let me tell you about the team that has worked on this project. Mira Garner was one of the main people who worked on the genetic data in the lab. She worked for us for about 6 months on this project, but had also worked at the Morton Arboretum previously on some oak work. She's now a graduate student at the University of British Columbia, so I think that having her work on this project really gave her some of the skills, confidence, and interests to go on to graduate school, and hopefully she'll take those forward for the rest of her career in plant ecology. Shown here are 3 other members of my lab: Emma Spence, Bethany Zimwalde, and Bailey Munoz, and each of them helped a little bit on this project in terms of organizing leaves or helping with some of the DNA extractions, etc. We had almost all the field work was done by collaborator Bill Carr, who is a famous botanist in Texas. He's really helped contribute to the knowledge of plant communities, plant biogeography where plants are in Texas, and also contributing to conservation. From what I found on the internet, he's collected more than 37,000 plant specimens to help us understand where plants occur. So he's a fantastic gentleman who was really helpful in our project, and he did basically all the field collecting, and maybe some of you know him. So what did we do in our project? In this project, we collected both from wild populations of big red sage and garden or nursery populations. So all those wild populations that were known that we could have permission to access were sampled by Bill Carr in 2018 and 2019. We also obtained samples from 2 nurseries and 1 garden, Ladybird Johnson Wildflower Center, to understand a little bit about the non-wild or ex situ plants. We also received seed from another nursery, and we tried to grow out the seedlings, but in spite of several different treatments, scarification, cold, warm, moist treatments, none of our seed germinated. And that's an important point that I'll come back to a little later. Seed germination is known to be not very good in this species. There are wild locations that we weren't able to access, including suspected locations of where the seed in nurseries originally came from. Just a little bit about the genetics. As you may read in the newspaper or hear about on the news, genetic technology has changed a lot in the past 5 years and is continuing to change. When we started this project, we had to choose what kind of genetic marker to develop. There's kind of more traditional genetic markers that people have been working with since the early to mid 1990s, so for more than 20 years. And they're quite cheap, pretty quick to develop, and we know them very well. Then there's what's called next generation genetic sequencing. And it is more costly. There are some trade-offs. It's usually not done in-house, so it's something we wouldn't have the technology to do all of it here at the Morton Arboretum. We would have to send that to a collaborator. I should mention we did, I forgot to acknowledge them, but the Field Museum of Chicago also helped with this project by allowing us to use their laboratory for some parts of this process. But anyways, we had to decide between this more traditional and newer generation of genetic technology. The more traditional technique is reliable, but a little less information. It's just not as powerful. And it's a hard decision and has been since 2015 and probably will be for the next 5 years. After, another 5 years, this more newer generation of genetic technologies will become more common. Something I want to note, I showed you this diagram of the different steps of the process of this kind of study. The genetic technology part is here, circled in red. All the other parts of the study remain the same and will remain the same for the foreseeable future. We still have to go out into the field. We still have to do some lab work, and we still have to interpret the results in the same way, no matter what technology we use. We went with microsatellites, which are the more traditional and much cheaper and more accessible genetic technology. This is a little technical and maybe not necessary for some of the audience, but when you're working with a rare species, you have to do what's called develop genetic markers. You have to take a piece of the DNA that's been really well studied in other species and then see if it can actually be applied to your species. Anytime you work with a new rare species, you have to go through that process. So we tried 100 markers from throughout the Salvia genus, which is really well studied because it's an important medicinal and horticultural species. So there's lots of not genetic knowledge in Salvia in general, just not in Salvia pentstemonoides. We got 26 of these to work, and we're feeling really good in our project about that. That's about what we expected. 12 of them were not very reliable though, and this happens with DNA because the DNA sequence itself, they just didn't amplify the same every time, which means they're not reliable data. Then 13 of the remainder were monomorphic, which means no difference among any of the plants that we examined, which was a little unexpected. And then only one was polymorphic. It seems to be amplifying at 2 locations, which can happen in the DNA, where you have the same sequence at multiple places in the genome. So we scored it as 2 what are called loci. in total, we looked at 171 plants of this rare species. What does the actual data look like? So here is shown the actual data, and it illustrates one of the challenges with working with really rare species. Because the populations are so small, the species might show little or no variation. And that's what you see here. If you go down this, each row is a plant. They're named garden or wild, and then each one has a population and then an individual number. As you go down this row, you see no variation in these numbers. That's what we call monomorphism or low polymorphism, and it just means we can't tell individuals apart. We can't get a good, unique DNA fingerprint per individual. There's a little variation here, and then this marker is very poorly amplifying. That's why it says no peak. So this one is not reliable, and we would skip that one. The rest are mostly monomorphic, which just means we can't get as much information as we would like out of these markers. So that's one of the common problems faced when working with rare species conservation genetics. Another common problem is accessing the plants. Rare species are rare because they're in difficult habitats, in unique habitats, hard to find, small, etc. So both finding the plants and accessing them was difficult for Bill Carr and his wonderful volunteers. So again, thank you to all the volunteers who helped. If some of you are on, really thank you very much for all your volunteer work in the field. This is an example. So having to use a ladder, first having to drag the ladder out into the field, and then use it to access these cliff faces. And then this is, you can see the two stalks here circled in red of Salvia pentstemonoides that were far out of reach over a cliff edge. So there were some individuals that were observed but could not be accessed. A common problem with rare plant studies. A third problem, which we didn't face as much, some plants have really tough leaves or lots of pubescence or harsh phytochemicals inside the leaves for defense. But that wasn't too much of a problem for us because we work on oaks and they have those same characteristics, so we solved those problems. So let's talk a little bit about what we found in our study. Like I said, we looked at cultivated sites, so gardens and nurseries, and we looked in the wild. We had a few more samples from the cultivated sites. These three rows are genetic statistics. What do they mean? Well, this tells us there was more variation, basically more genetic diversity found in our wild sites, which is not a surprise because anything in cultivation originally came from the wild, so it's some subset. But this is how we can summarize our data at a really high level. So that's what I just said. What is currently ex situ in gardens and nurseries is a subset of the wild, a little over about half of what we found in the wild. Like I said, we saw very low polymorphism. This was unexpected, not totally unexpected because it's a rare species, but a little bit unexpected, much lower than we expected. It could signify that these populations have very little genetic diversity and would not be able to adapt in the future. But this could be a characteristic of these genetic markers, and I'll come back to that in the future directions. Like I said, it's higher in the wild, but it's still low. So especially this heterozygosity, we would normally want that to be up in 0.6, 0.7 or higher. So this is quite low, but is not uncommon for a rare species. So another thing we found was some of the gardens and some of the wild populations had more diversity than others. In particular, one of the nursery populations had very low, almost no diversity, and same for some of the wild populations. There are anecdotal reports of very low or no seed set in gardens and low seed viability from wild populations. Seed that we got from a nursery did not germinate. Could have been just that batch, or it could have been what we call inbreeding depression. So this low diversity in some populations, including some nursery centers, might be leading to lack of viability in seed in some cases. This is a picture of our results. So this is everything I just told you in a picture form. The 3 cultivated populations are on top, and the 4 wild populations are on bottom. Each color is a unique genotype. Immediately you see each wild population is very different from each other. So each of these wild populations has unique diversity and is worthy of conservation. The gardens are different from each other, but more similar to each other than the wild populations are, probably because they share some of the first original seed source. One of the gardens clearly has one of the nurseries has very low genetic diversity. Each one does different from each other, but looking at these shared colors, it could be they all came from the same original site. So let me summarize all that again. The wild populations have low genetic diversity, emblematic of rare species. Each wild population is different and valuable genetically. The cultivated populations have even lower genetic diversity than the wild. They each do differ from each other in genetic composition, but they are similar enough they could have come from the same seed source. Again, overall, both in the wild and cultivation, the species genetic diversity is quite low. And because we have small population sizes, in some cases just 2 or 4 or 10 plants were found, and so genetic diversity is likely to keep degrading in the wild. Some of the wild genetic diversity is not protected ex situ, so a lot of these colors are not found in the gardens and nurseries. If seed collection is to occur for botanic gardens or nurseries, seed collection should be done by conservation professionals to ensure 4 criteria: not damaging the wild populations, so not damaging those highly erodible, fragile habitats, not taking too much seed. Seed should remain in the habitat for future regeneration of the plants. We do need data taken about where the plants come from, how many plants there are, and we do need to be able to try out different germination and protocols to make sure the seed survives. What are some actions that could be done in the future? One is because these populations are so small, genetic diversity will continue to disappear unless the population numbers increase. This could be done by improving the habitat or actual planting of individuals from gardens or planting seed collected in the wild. Typically, at least 50 individuals are needed to prevent inbreeding, and more than 500 are needed for that population to be able to adapt to ongoing environmental change. We weren't able to include all the wild populations, so this study could be expanded in the future to get more understanding of those wild populations. There does seem to be a lot of variation anecdotally in how many plants are observed in a given year. So some plants might not flower in a given year and might not be found. Some plants or population sizes might actually be shrinking or growing. So regular surveys are needed to check for that. Because these populations are so small, it may help to move seed among the populations, but we can't say that for sure based on our data yet. We don't know if this is necessary or the best idea. My suspicion, based on my work in other rare plants, is that this could help the genetic health of these populations, but that's not confirmed yet. And then lastly, because there are so few populations, we do need more populations established to improve the overall status of this species. So other rare plants like the Tennessee coneflower or the eastern prairie fringed orchid, which is found in the upper Midwest, these are other species that have shown to have low genetic diversity in few populations, but have been recovered to a large extent, really good conservation successes in spite of their few populations and low genetic diversity. So again, like I said, repeated surveys over time would be useful, increasing the presence of this species in botanic gardens, perhaps moving seed or pollen among them to have more genetic diversity. And then, like I said, we're at this transition with genetic technology, and the next generation sequencing approach could provide finer resolution and more detailed results than we were able to find in our study. Overall, I hope you've seen that it's useful and exciting to study rare plants with conservation genetics, and that it has taken a lot of partnerships between the Botanic Garden, volunteers, professional botanists, nurseries, and many other conservation actors. So thank you in particular to Texas Parks and Wildlife for funding this through the Conservation License Program. For those of you who have conservation license plates, thank you. You helped fund this. Thank you to the Morton Arboretum, which also provided supplemental funding, including my own salary, but also supplies. Some of my other work has been funded by the Institute for Museum and Library Services. Thank you to the Center for Plant Conservation, which has been a consistent advocate for rare plants and has helped promote and support these kinds of studies. And to my lab members, I forgot to put a picture of Mira Gardner and a thank you to the Field Museum as well for letting us use their facilities. And thank you all for listening today. I hope it's been enjoyable and useful.
Thank you, Sean. We do have some questions. The first question is, was there much plant litter where the plants were growing in the wild, or is it almost bare rock? Do you want me to answer that one, or do you want to?
I think you might have a little more familiarity. I should say that I was not in the field myself, so the people most familiar would be Anna Strong or perhaps other people who are on the call.
Yeah, so I mean, it varies a little bit between sites, but as you saw on the pictures that Sean showed earlier, most of the populations are on bare rock or even bare rock cliff faces. So yeah, that's the most common habitat. The next question is, what is their chromosome number? Are they polyploid?
So the chromosome number, I used to have I've been cleaning my office. I used to have a stack of papers, so I did some research into this before we started the project. We wanted to make sure it was feasible. And so I looked at chromosome numbers throughout the Salvia genus. There are a couple of such publications. If you'd like to know precisely, just email me. My email is shoban@mortonarb.org. Sorry, I forgot to put it on here. I can look that up. There are some publications, and we did look it up. I want to say 22, but that is a bit of a guess from a year and a half ago. As for polyploidy, some Salvia are polyploid. There's it's associated with the evolutionary radiation of the species, which was a whole other fascinating thing to read about as we prepared for this project. The subsection that this species is in, those species that have been studied seem to be mostly diploid. So that's whether they have one set of chromosomes or multiple sets of chromosomes. For the non-geneticists, basically the size of their genome, and the number of genetic marker copies that we would see. So to my knowledge, based on the research we've done, they're likely diploid, and I can send any papers for those who are interested.
The next question is, first, how far apart are the wild populations? And second, can you enhance gene flow between wild populations through pollen transfer between sites? I'm not sure how much to talk about the wild populations, locations. Do you want to take that one? Yeah, so I can't remember the exact distance between the populations that were collected from, but let's just say it was across 1 or 2 counties, and these are hill country Texas counties. So it gives you an idea.
Yep. That's my impression looking at the map as well. As for enhancing gene flow through pollen transfer, I think it would be best to experiment before actually trying it. It's not known. It's done in some species. So our plant breeder transfers pollen all the time from one plant to another, including across the site. It can be viable. Depending on the species, it can be viable for minutes, hours, or days. So there's a really big dependence for pollen viability. So I can say maybe to that. Usually when we want to ask that kind of question in a rare species, we will first test that in a more common species. So we would maybe want to try that in a more common salvia first, perhaps in a botanic garden center to see if it works. And then it could be tried in this rare species.
So the next one I think is more of a comment, but it says, in the hill country, the big problem is goats, sheep, deer, and hogs, and plants have to be inaccessible even to a goat. That is true.
Yeah, and it's possible this species was more common in the past, and only those very inaccessible plants have survived. Definitely possible.
Was any information found that explains the unique chemical smell of the leaves?
So we didn't study that in our project. We didn't look at the genes that might underlie different traits like floral scent. So I can't answer that one. We don't have knowledge on what causes the floral scent.
Next question, where was diversity greatest among botanical gardens?
So there was pretty high genetic diversity found in the Lady Bird Johnson Wildflower Center and in one of the nurseries. And one of the other nurseries had quite low genetic diversity. We would love, I mean, in the future that we could also study other botanic gardens. Our project was a bit limited in time and scope, but one could look at other botanic gardens in the future.
Is there a way to tell the difference between inbreeding depression and local adaptation in your results?
That's a really insightful question. Both might cause low genetic diversity. Adaptation does mean that some individuals are eliminated from a population, those that are not adapted, which could result in low genetic diversity. I think the major difference, there's a couple differences. We wouldn't expect that to happen at every genetic marker in that case, only those underlying the local adaptation. So we would see like some markers that have a lot of diversity and some that have low. And then inbreeding would always result in low heterozygosity. So that's all the kind of copies in a given individual being the same. So there are ways by looking at multiple of these different statistics and lots of genetic loci. But you're right, those could be a little bit similar.
What exactly does no peak mean for the data?
Sure. The way we examine this genetic type of marker, we look at basically what's called a trace. It's a screen where you can see me. There's a peak when you see the actual genetic locus of interest. And so no peak means it didn't show up. It basically means that piece of the DNA was hard to amplify or hard to examine, and we just didn't find it on that individual. So when there's lots of those no peaks, it means it's not easy to look at the DNA of that part of the DNA.
I purchased a plant from Lady Bird Johnson Wildflower Center a few years ago, and I have been letting it naturalize. I have seed collected for the past 2 years. Do you have a need for samples from people's home gardens, either just for study or possibly to help contribute to gene pool?
That's a really good question. Maybe Anna, do you want to take a first stab at that, and then I can follow up?
I mean, at this point, we don't have plans to fund any future projects with Salvia penstemanoides simply because of other priorities. But for TPWD specifically, we don't have a seed bank. It is possible that Lady Bird Johnson Wildflower Center, who has a seed bank, would need or want your seed, but we don't have a facility at TPW to take it.
Okay. We also don't actually have a seed bank. Our main focus at the Arboretum is living plants. From what I've read, Salvia penstemanoides would probably not survive here in Chicago. So in terms of need for a seed, one could reach out to some of the biologists at Lady Bird Johnson Wildflower Center and see what they think, if they are interested in holding that seed in the seed bank or in distributing it to people nearby.
The next question is, did y'all observe any wildlife interactions, for example, pollinators? And unfortunately, none of the people that were actually in the field for this particular study are on the phone, although a couple of them have responded with black-chinned hummingbirds and a porcupine. So hopefully that answers your question. The next question is, how about bottlenecks in the past to account for low genetic diversity?
Great. I see a couple of people chiming in work here. So again, for those who volunteered and participated in those field trips, I hope it was enjoyable and useful, and thank you for your help and volunteer hours and commitment to this species. And feel free to type into the chat if there's anything more you have to say about what you observed in the field. As for bottlenecks, certainly there's been, I think there's been some kind of bottleneck in what we see in the DNA and in how many plants we see in the field. The question is, how far back in the past does that go? So I think this question might refer to, is it like a long time ago or more recent? From what we know about habitat loss, from what we know about herbivore problems, and from what we know about the species being well known and then kind of being presumed extinct, it's probably a more recent bottleneck that has caused the low genetic diversity.
Is there a concern with nursery populations being artificially selected for some desirable trait and then those genes entering wild populations to a negative effect?
That is always a tricky conundrum and concern in plant conservation. So yes, it's certainly a concern. Do we know that it could be a problem? I think that's less sure. It is known that when you grow rare plants in cultivation, you might be selecting for certain traits which might not be adaptive in the wild, such as really large plants when in the wild they need to conserve resources, or too many flowers, or flowers that flower at the wrong time, or things like that. So it's certainly a concern in any rare species being grown in cultivation. On the other hand, sometimes growing it in cultivation can save genetic diversity or even the whole species that would go extinct. So there's a trade-off there, and I can't say that there's one best answer to that.
Somebody from the Wildflower Center has said, for those of you who have seed and would like to donate them to the center, just get in touch with them via the staff contact on their website. Also, for those of you who want to see pollinators of Salvia penstemanoides, at least in Travis County, can visit the plants at the Lady Bird Johnson Wildflower Center. Somebody has asked if the talk is being recorded, and it is. It will be loaded on the same website, which, let's see. assuming I have control, I can share my screen one more time and show the URL for those of you who want to be reminded of where to go to register for future talks. Are there any other Texas nurseries besides the Wildflower Center that sell this plant? And the answer is yes, and you can find them online. One nursery owner has put his information in the chat, and that is at Natives of Texas Nursery in Kerrville. You can find that online, nativesoftexas.com. There's also a nursery in Medina, Texas called Medina Garden Nursery that you can find the plant. There's a question. In the structure diagram, was each bar an individual plant with colors representing unique genotypes?
The answer to that is yes. So that's this plot here. For those who are...
Sorry, Sean, I was in the middle of...
Okay.
Hopefully people saw it for a second there because I was about to show the last slide. Sorry about that.
Okay, no problem. So the answer is yes. Each of those is a unique individual, and that's what each of those colors represent. I should mention that this is a unique genotype, but this is based on only 2 genetic markers that were polymorphic. So there are probably more unique genotypes, but that's all we could tell. Are you still there?
Yeah, I am. Sorry. I was just making sure there were no more questions in the chat.
Okay, because my computer froze, so I just wanted to make sure you're still there.
Another question is, so then was a W1 a possible source for a nursery population? And the answer is no, that was a wild population, right?
Yeah. So I can't see my slide at the moment, but yeah, we had wild populations on the bottom and nursery populations on the top of that diagram. And it's hard to say because the sample sizes are a little small, but one of those wild populations would have matched the nursery populations better. But there are a few wild populations, at least one known, that we were not able to access. So it could have been another location.